Why does adrenaline, also known as epinephrine, constrict some blood vessels and dilate others? Why do beta blockers slow the heart but can also worsen asthma? It comes down to 4 adrenergic receptors, and once you know them, everything clicks. In this video, we'll break down the 4 major adrenergic receptors, where they are found, what they do, and how they explain the actions of key drugs. If you've seen my autonomic nervous system lecture, this builds directly on that. Before we dive into adrenergic receptors, let's clear up a naming issue, because textbooks often use 2 different pairs of terms. Most of the world says adrenaline, since it comes from the adrenal gland. Logical, right? In the US, especially in pharmacology, the same molecule is called epinephrine. Chemically, they're identical. The difference is purely historical and linguistic. The reason for the split is surprisingly silly. Around 1900, the word adrenaline was trademarked by a pharmaceutical company, so US naming committees introduced epinephrine, and that name stuck in American drug labeling and pharmacology. Physiologists tended to prefer adrenaline, while US pharmacologists leaned toward epinephrine, leaving generations of students confused. For consistency in this lecture, I'll use adrenaline. Just remember, adrenaline equals epinephrine, noradrenaline equals norepinephrine. The only difference is naming tradition, not chemistry. With that sorted, we can move on to adrenergic receptors and explore how this single chemical family produces such a wide range of physiological effects. Adrenergic receptors, or adrenoceptors, are G-protein coupled receptors that protrude from the cell membrane into the extracellular space. They have specific binding sites for catecholamines, which activate them to stimulate signaling pathways in the cell and produce a response. There are 2 such molecules produced physiologically. Noradrenaline is released from the terminals of sympathetic postganglionic neurons innervating tissues. Adrenaline is secreted from the adrenal gland medulla into the circulation, which carries it around the body to act on distant adrenergic receptors. Adrenergic receptors are divided into alpha and beta subtypes, each with characteristic signaling pathways that explain their physiological effects. Let's start with the simplest way to organise them. All adrenergic receptors are G-protein coupled. α-adrenoceptors couple with GQ, α-2 receptors couple with GI, and all the beta receptor subtypes couple with GS. This allows them to modulate cell signaling pathways in different ways, and understanding how they do that helps to predict their actions on the tissues in which they are expressed. Knowing which G protein each subtype couples to makes the physiology much easier to predict. For example, the GQ protein enhances calcium signaling, which causes smooth muscle to contract, leading to constriction of smooth muscle organs such as blood vessels, the iris dilator, and sphincters. In glands like the salivary and lacrimal glands, increased intracellular calcium stimulates secretion. GI proteins tend to inhibit calcium signaling in neurons, so α-2 adrenoceptors located on presynaptic nerve terminals reduce the calcium influx that normally triggers neurotransmitter release. The same mechanism explains why alpha-2 receptors inhibit insulin release in pancreatic beta cells. Reduced calcium entry means reduced exocytosis. GS proteins tend to have the opposite effect to GI, but their downstream actions depend very much on the cell type. For example, in cardiac muscle, GS signaling enhances calcium handling and increases the strength of contraction, whereas in smooth muscle, it suppresses calcium signaling and promotes relaxation. This apparent contradiction occurs because G proteins do not usually control calcium influx directly. Instead, they modulate intracellular second messengers, which then influence whichever calcium regulating mechanisms the particular cell expresses. As a result, the same G protein pathway can produce very different physiological outcomes in different tissues. Let's follow these pathways in more detail to see why the same G proteins can produce such different effects. The G in G protein stands for guanine nucleotide. These are guanine nucleotide binding proteins made up of 3 subunits: a single alpha subunit and a beta-gamma complex. In its inactive state, the alpha subunit binds GDP, and all 3 subunits remain bound together. When the subunits are together like this, the G protein can interact with an adrenergic receptor. When adrenaline or noradrenaline, the agonist, binds to the receptor, its shape immediately changes. This new conformation causes the receptor to interact with the G protein, stimulating it to release its GDP in exchange for a GTP molecule. This exchange activates the G protein, causing the alpha subunit to separate from the beta-gamma complex. Both activated parts are then free to move away and interact independently with downstream signaling molecules to change cell behavior. Meanwhile, the agonist can dissociate, allowing the receptor to return to its original shape, ready for the next signal. The alpha subunit has built-in timing. It's a GTPase enzyme. This intrinsic activity eventually hydrolyzes or breaks down the bound GTP back into GDP. Once the GTP is converted back to GDP, the alpha subunit loses its activity and recombines with the beta-gamma complex. The entire G protein is now inactive, ending the cell signaling effect until the next agonist binds. It's the alpha subunit of a G protein that determines which signaling pathway will be modulated. So the alpha subunit effectively gives its name to the G protein. GQ uses alpha Q, GI uses alpha I, and GS uses alpha S. The beta-gamma subunits, by contrast, are largely conserved and not specific to any particular G protein class, so the major functional differences arise from the alpha subunit. Starting with GQ, the alpha Q subunit activates the enzyme phospholipase C, which splits the membrane lipid phosphatidylinositol 4,5-bisphosphate into 2 second messengers, inositol 1,4,5-trisphosphate and diacylglycerol. IP3 triggers calcium release from the endoplasmic reticulum, while diacylglycerol and calcium together activate protein kinase C, which phosphorylates a range of cell proteins. This pathway is central to calcium-dependent responses in the cell, leading to contraction and secretion. The GI and GS proteins both modulate the same target enzyme, adenyl cyclase. But while GS stimulates it, GI inhibits it. Adenyl cyclase converts ATP into cyclic AMP, which then stimulates protein kinase A to phosphorylate a wide range of different proteins. So, while GS stimulates cyclic AMP production and downstream phosphorylation, GI dampens responses that would normally be triggered by cyclic AMP. So, although adrenergic receptors all activate G proteins in a similar way, the alpha subunit differs and determines which effector pathway is engaged and shapes the cell's physiological response. Now let's look more closely at each of the receptor subtypes, starting with the alpha-adrenergic receptors. There are two main subclasses, alpha-1 and alpha-2. Both respond to noradrenaline and adrenaline, but they couple to different G proteins and have distinct effects. Alpha-1 receptors are coupled to GQ proteins, so activation triggers the phospholipase C pathway, increasing intracellular calcium. We typically think of alpha-1 receptors in smooth muscle, where they cause contraction. That results in the constriction of blood vessels and raised blood pressure. Contraction of the iris dilator muscle, leading to pupil dilation, and bladder sphincter contraction, which promotes urinary retention. But alpha-1 receptors are also found in exocrine glands, particularly the salivary glands. Activation here stimulates the fluid component of secretion and contributes to the overall thick, viscous saliva seen during sympathetic nerve stimulation, which is due to the balance of alpha-1 and beta receptor effects. It's a secretion quite different from the watery one driven by parasympathetic stimulation. Clinically, alpha-1 agonists like phenylephrine are used primarily for their effects on blood vessels. By constricting small blood vessels in the nose, they are effective decongestants. They may also be used to raise blood pressure in hypertension. Antagonists at alpha-1 receptors reduce sympathetic constriction of blood vessels and are used to treat hypertension. They also reduce the alpha-1 mediated stimulation of prostate smooth muscle by the sympathetic nervous system, making them effective treatments for benign prostatic hyperplasia. Alpha-2 receptors couple to GI proteins, which inhibit adenyl cyclase and decrease intracellular cyclic AMP levels. They are located predominantly on presynaptic nerve terminals in both the central and peripheral nervous systems. They act as autoreceptors on sympathetic nerve terminals to inhibit noradrenaline release and provide an important negative feedback mechanism. Alpha-2 receptors are also found on parasympathetic nerve terminals, where they function as heteroreceptors and modulate parasympathetic neurotransmission. Clinically, alpha-2 agonists such as clonidine lower blood pressure, primarily through a central action that reduces sympathetic outflow, and they are also used for sedation. Alpha-2 antagonists like yohimbine are rarely used, for example in erectile dysfunction. Mirtazapine is a non-selective alpha-2 antagonist. Blockade of these receptors, together with its serotonergic and histaminergic actions contributes to its antidepressant effect. Overall, you can think of alpha-1 receptors as stimulatory and alpha-2 receptors as inhibitory. Now let's look at the beta-adrenergic receptors, beta-1, beta-2 and beta-3. Although all three couple to GS proteins and increase intracellular cyclic AMP, their tissue distribution and physiological roles differ quite a bit. Starting with beta-1 receptors. These are concentrated in the heart and in the juxtaglomerular cells of the kidney. Activation increases heart rate, contractility and conduction velocity through the heart. In the kidney, beta-1 stimulation triggers renin release, recruiting the renin-angiotensin-aldosterone system to provide sustained support for blood pressure and blood volume, extending the influence of beta-1 activation beyond the immediate cardiac effects. Beta-2 receptors have a broader distribution. They are found in the smooth muscle of bronchi, certain vascular beds, and the uterus, where they promote smooth muscle relaxation and dilation. In hepatocytes, they stimulate glycogenolysis. These effects become particularly important during exercise and stress, when increased airflow, energy mobilization, and redistribution of blood flow are advantageous. Finally, beta-3 receptors are located mainly in adipose tissue, where their activation promotes lipolysis. They are also expressed in the bladder detrusor muscle, where alongside beta-2 receptors they contribute to bladder relaxation. Across all three receptor types, the common theme is activation through GS proteins and cyclic AMP, but with distinct physiological outcomes determined by the target tissue. Let's talk about the clinical uses of drugs that target beta-adrenergic receptors. Beta-1 receptor agonists, like dobutamine, are used in acute heart failure or cardiogenic shock because they increase cardiac output. Beta-2 receptor agonists, like salbutamol and salmeterol, relax bronchial smooth muscle, which makes them essential for asthma and COPD. Even though beta-2 receptors exist in other tissues, inhalers localize the effect to the lungs. They are also used as tocolytics to relax the uterus in preterm labour. The beta-3 agonist mirabegron causes detrusor relaxation to treat overactive bladder, and because beta-3 receptors have a limited distribution, it lacks the side effects that a beta-2 agonist would have. Now moving on to beta blockers. The beta-1 selective blockers, like atenolol and metoprolol, are commonly used to treat cardiac arrhythmias, heart failure and hypertension because they reduce heart rate, contractility and oxygen demand while being relatively sparing of beta-2 receptors in the lungs. These are the cardioselective beta blockers. Non-selective beta blockers, such as propranolol, block both beta-1 and beta-2 receptors. They help with heart conditions such as angina and hypertension and are used for thyroid storm, migraines, essential tremor and some anxiety states where there may be hyperactivity in the sympathetic nervous system. When used for glaucoma, they inhibit beta-adrenergic stimulation of the ciliary muscle in the eye, while delivery in eye drops limits its spread to the systemic circulation. But remember, beta-2 blockade can trigger bronchoconstriction in sensitive patients. Finally, non-selective beta blockers with alpha blockade. Drugs like carvedilol and labetalol combine beta blockade with vasodilation from alpha-1 inhibition. This makes them particularly useful in heart failure and hypertensive emergencies. So, while all these drugs target beta receptors, their selectivity determines whether they mainly affect the heart, lungs, blood vessels or other organs, and that shapes their clinical use. Noradrenaline and adrenaline are structurally related, but distinct molecules. It's therefore not surprising that they interact differently with adrenergic receptors. They each have a different selectivity profile for adrenoceptor subtypes. This is the order of potency for noradrenaline. It strongly activates alpha-1 and alpha-2 receptors, moderately stimulates beta-1 receptors, but it is weak at beta-2 and beta-3 receptors. Because alpha-1 receptors dominate in the vasculature, noradrenaline causes widespread vasoconstriction when released from sympathetic nerves. That raises peripheral resistance and blood pressure, and any direct beta-1 mediated stimulation of the heart is often masked by reflex slowing of the heart. Its weak action at beta-2 and beta-3 receptors means noradrenaline produces little bronchodilation and minimal metabolic or bladder effects. Adrenaline has quite a different pattern. It strongly activates beta-1 and beta-2 receptors and only moderately activates alpha and beta-3 receptors. Adrenaline therefore increases heart rate and contractility through beta-1 receptors, and its strong beta-2 activity produces bronchodilation and vasodilation in skeletal muscle. These beta-2 mediated vascular effects reduce total peripheral resistance, which is very different from the pressor response caused by noradrenaline. Adrenaline also drives metabolic responses, such as glycogenolysis and lipolysis, again through beta-2 receptors. At higher concentrations, it can also stimulate alpha-1 receptors and cause some vasoconstriction, but beta receptors dominate the response. So, although both catecholamines act on the same family of receptors, their differing affinities mean they produce quite distinct physiological responses. Here's a quick overview of the adrenergic receptors and why they matter clinically. Alpha-1 receptors couple to GQ proteins. They contract smooth muscle, causing vasoconstriction and helping to raise blood pressure. Alpha-2 receptors coupled to GI proteins reduce sympathetic outflow by decreasing noradrenaline release at nerve terminals. Beta receptors all couple to GS proteins. Beta-1 receptors are mainly in the heart, increasing heart rate, contractility, and renin release. Beta-2 receptors relax smooth muscle, producing bronchodilation and vasodilation. And beta-3 receptors relax the bladder detrusor muscle, as well as causing lipolysis. Clinically, we use these receptors in targeted ways. Beta-1 agonists support the failing heart in shock. Beta-2 agonists are essential in asthma and COPD and can also relax the uterus. Beta-3 agonists, like mirabegron, treat overactive bladder. On the antagonist side, beta-1 selective blockers treat heart disease and hypertension with less effect on the lungs. Non-selective beta blockers have broader uses, including tremor, migraines, anxiety, and glaucoma. And combined beta and alpha blockers, like carvedilol, are especially valuable in heart failure and hypertensive emergencies. So The key idea is that receptor selectivity guides where these drugs act, whether on the heart, the lungs, the vasculature, or the bladder, and defines their clinical roles.